Okay, welcome everybody. This is chapter 24 dealing with metabolism, which is the back end of digestion. Essentially, the digestive process is to chemically and physically break down food into its simplest building blocks. The proteins are broken down into amino acids, the complex carbohydrates are broken down into simple sugars, and the fats are broken down into fatty acids and glycerol. And in that form, it's loaded into the blood. Okay. Now, metabolism addresses what happens after it's in the blood and ends up being delivered to the cells in order to convert the power that's in those building blocks into a form that the cell can use, which is ATP. Okay. Ingested nutrients can be used for two reasons: to generate fuel for the cell to maintain itself and repair itself and to grow, but also as building blocks for other cellular components. The energy value in these fuel molecules is measured in kilocalories, which is defined as the heat needed to raise the temperature of a kilogram of water by 1 degree centigrade. These absorbed nutrients follow different fates once they move across the cell membrane into the cells that make up our tissues. A nutrient is any substance that we need for growth, maintenance, and repair. The major nutrients are the bulk of the food that we eat, and those are carbs, lipids, and proteins, and they contain chemical energy in their chemical bonds that can be liberated when those bonds are broken. And then that energy can be moved over to ATP, with some energy lost as heat in the process. Other nutrients are needed in small amounts, and those include vitamins and minerals. The primary use for vitamins and minerals is to aid in enzyme activity in order to carry out the chemical reactions that support life. Water is technically a nutrient because it is an environment in which the other chemical reactions happen. It's also a participant in and a result of various chemical reactions. The food groups are fruits, vegetables, grains, protein, and dairy, and all of these consist of differing amounts of calories per gram. We want to keep our calorie consumption around 2,000 calories a day to maintain our body weight for a healthy individual in the prime of life. So that would be a young adult. Okay, and you would adjust those numbers depending on your age and your sex. If you eat more than that and you don't burn those calories off, then you're going to gain weight in the form of fat. And if you eat less than that and burn more than those calories off, then you're going to lose weight. We want to get plenty of fruits and vegetables and whole grains, and we want to avoid food that's high in calories, and we want to exercise. And the reason for that is we want to cut down on the amount of adipose tissue that the body stores. Um, the storage of adipose tissue is really an evolutionary adaptation to a time when we didn't eat regularly. A long time ago, we used to live in groups in caves, and we would eat whatever walked past the cave. And that might serve as your only meal for many weeks or months. And if the body didn't have the ability to store some of those calories as fat, you might not be able to live long enough to make it to your next meal. And that's the reason also why we like the taste of things that are high in calories. That's why we like sweet things and we like things that are meaty or high in fat. And we tend to dislike things that are bitter. Those things happen to be poisonous. And so we've been naturally selected to have an aversion to that flavor. Okay? And that's reflective of, again, a survival behavior. You can see the food pyramid here. The food pyramid is thought to be a little bit outdated. I don't really think it is. But the idea with the food pyramid is that the low calorie stuff is at the bottom and the high calorie stuff is towards the top. Okay? And so you can see the number of servings for each. and when to eat each. The bottom line is that if you want to maintain a healthy body weight, you have to burn as many calories as you consume. A simplified version of the food pyramid in use now is my plate, which basically uses the surface area of the plate in order to indicate how much of each of the food groups you should consume at a typical meal. Okay, um, the different colors are reflective of some of the dominant colors in these different food groups. Okay, with the exception of the protein being purple. Now, an interesting thing about your body is it has the ability, under certain situations, to convert certain nutrients into other molecules that can be used to generate energy, or molecules that can be used to manufacture things like proteins and polysaccharides. The liver does a lot of this um, in processes known as transamination and deamination. The liver can create amino acids, and it can convert amino acids into intermediates that we normally see in pathways like glycolysis and the Krebs cycle, which is designed primarily to produce energy for the cell. Okay, so in a pinch, the body will go to our protein in order to generate energy if we don't have a store of fat or glycogen to utilize. Okay, but the primary reason that you eat protein is to make protein when you're eating a balanced diet, right? So we break down the proteins in our diet, we strip out the amino acids, and then we reassemble them into the amino acids that carry out the work of life in our own body. Essential nutrients are the ones that we have to eat in order to have. The liver can't make these. There's about 50 essential molecules, including essential amino acids and fatty acids, that we have to consume in order to have. Non-essential nutrients are also critical to life. I mean, at the bottom line is we are what we eat. Okay, and if we don't eat enough to support the maintenance of the body, then we're slowly going to starve to death. Carbohydrates are an example of a high-calorie group. Dietary sources come mostly from plants. Starch, which is a complex carbohydrate, is found in grains and vegetables, and also to some extent in fruits. Sugars are found in fruits, sugar cane, sugar beets, honey, and milk. Fiber is really in all vegetables and fruits. Insoluble fiber includes the cellulose in vegetable and provides roughage and bulk to our diet, while soluble fiber includes pectin in apples and citrus fruits, which can aid in reducing blood sugar and cholesterol levels. And there's a small amount of carbs in milk, and of course there's some glycogen in meat. Now, the bottom line with carbohydrates is that they're water soluble, okay, and they are a calorie source with the exception of cellulose and hemicellulose, which we don't have the ability to break down and strip out the energy from those chemical bonds, okay, because we don't have the enzymes to do that. And so that passes through our digestive system as fiber and out the other end. Um, the sugars have a lot of carbon hydrogen bonds in them and a lot of carbon hydroxide bonds in them, and those contain a lot of energy. And so when you break those bonds, the energy that's released is used to manufacture ATP, and that can be used by the cell to do work immediately. Okay, and then a considerable amount of it is lost as heat. Okay, other uses for carbohydrates, um, other than using it as fuel to drive ATP production, um, includes the use of carbohydrates as backbones for nucleic acids like DNA and RNA. It's one of the building blocks of nucleotides like ATP. Okay, um, and then it's also used to generate glycoproteins and glycolipids, which can serve as identifying molecules for the immune system in our body. Um, and it serves as an energy storage unit in the form of glycogen, which is manufactured by the liver and our skeletal muscle. Okay. Um, if we take in more glucose than we burn, we convert that to glycogen, or we can convert that to fat and store that in the adipose tissue in our body. Okay. By the same token, if we need to put glucose in the blood, the liver has the ability to take that fat and convert it into glucose in a process called gluconeogenesis, which literally means to make new sugar. Okay. Fructose and galactose are converted into glucose by the liver before they enter the circulation, and in that form, the cells can take them up and use them for energy. Dietary requirements: minimum of 100 grams a day to maintain blood glucose levels, recommended 130, and that's to drive nerve activity, okay? Because the only fuel that the nervous system can utilize is glucose. Other tissues can use other fuel sources, and so that property is called glucose sparing. We always work to make sure that any glucose in the diet has as its first priority to go to power nerve activity, and then any remaining glucose can be used for other tissues. Recommended intake: about 45 to 65% of your total calories, as mostly complex carbohydrates, which we get from whole grains and vegetables. If you're less than 50 grams a day, then we go to protein and fat for energy. An example of this is the Atkins diet, in which we reduce carbohydrates dramatically and we eat primarily fat and protein, and the body then is going to break down our fat and use that for energy to drive our normal processes. But as a result of having so few carbohydrates in our system, we actually enter a condition known as ketoacidosis, in which some of the breakdown products of the fat become ketone bodies, which are eliminated in our sweat, in our urine, and in our breath. Okay, and so that's why in a lot of these diets, they start you out on something called induction. They send you into ketosis, and then they slowly reintroduce carbohydrates in a stepwise fashion. In order to maintain the weight loss, though, they keep the carbohydrate levels low. But if you go no carbohydrates for an extended period of time, you go into ketoacidosis for too long, that's going to cause a shift in the body's pH that can lead to coma and death. All right, and so that's not something that you want to end up doing. The American diet is about 46% carbohydrates that we get from rice, pasta, and breads. Highly refined carbohydrates from sugary foods can lead to nutritional deficiencies and a problem that's rampant in the U.S., which is obesity. And the reason that our diet tends to make us fat is because we have a lot of foods out there that are high in calories and low in satiety, meaning you can eat a lot of calories but not feel really full. And so the way around that is to always try to include in your diet a lot of high fiber material in order to make you feel full but not increase your calorie intake. Okay, so you get that primarily from your fruits and your vegetables. And the other thing to do is to go high protein, and that also makes you feel fuller but has a lower calorie content than if you eat something that's high in fat or high in carbohydrates. Lipids are the calorie champ, and the reason for that is that if you look at the structure of a typical lipid, it has about 20 times more carbon-hydrogen bonds than a typical carbohydrate or even complex carbohydrate. And so that's why if you eat a lot of fat, you don't do a lot of exercise, you're going to gain weight. Okay? Triglycerides are found as saturated fats in meat, dairy, and tropical oils. Unsaturated fats are in seeds, nuts, olive oil, and vegetable oil. We also get cholesterol from meat and dairy, okay, as well as shellfish. All of these are important in both structural components in the body as well as a source of energy. So the triglycerides are high in calories. So we can take the chemical bonds, the carbon-hydrogen bonds in the um, in the lipid, we can break those down, and a lot of energy gets released to support ATP production. In addition, however, triglycerides are the starting material for phospholipids, which are a major component of cell membranes, and they also serve as membrane components in the membranous organelles in the cells and the tissues. So that's their structural role. The cholesterol is utilized to control membrane fluidity, so it acts kind of like a doorstop in a typical phospholipid bilayer. You stick the cholesterol in there, and the phospholipids don't move around as much, right? It's also used as starting material for a variety of hormones like testosterone, estrogen, aldosterone, cortisone, cortisol, okay? And it's also an important component of bile salts, okay, which are used as an emulsifying agent in the digestive system. The liver makes about 85% of the cholesterol despite our dietary intake, and it can produce it from other sources. The liver can convert fatty acids to each other. However, there are essential fatty acids that we have to eat in order to have, and those include linoleic and linolenic acid, which are found in vegetable oil, so they have to be eaten in order for us to have them. Lipids help us absorb fat-soluble vitamins. Those are A, D, E, and K. And it's a major fuel for liver cells and for skeletal muscle. We've talked about phospholipids in cell membranes and membranous organelles. It's also critical in the production of the myelin sheaths in the central and peripheral nervous system. And adipose tissue, of course, is used for protection and insulation as well as fuel storage in the body. Prostaglandins are derived from fatty acids, and they cause smooth muscle contraction. They aid in the regulation of blood pressure, and they're also critical intermediates in inflammation. Okay? And we've talked about the uses of cholesterol. Okay, dietary requirements for lipids. Fat should be 30% or less of your total calorie intake. Saturated fat, 10% or less. Cholesterol, no more than 300 milligrams daily. The goal is to keep your cholesterol below 200 milligrams per deciliter, and your typical American diet, greater than 40% of our total calories come from lipid, okay? And so again, this is one of the reasons that we tend to be obese, right? We have a lot of components in our diet that are high in calories, but they don't really fill us up very well, okay? Fat substitutes include air, which can lower the density of food and make it take up more space, modified starches and gum, whey protein, olestra, a P&G product, which is an indigestible fat, and gum derivatives and olestra, we should note, are not metabolized. They pass right through the digestive system and out the back end. The rest of these are metabolized and result in calories. The disadvantages of fat substitutes is that they usually aren't good to cook with. The taste differs from real fat. They can cause gas and diarrhea because when they get to the colon, the bacteria there can break down some of these fats and generate gas as a byproduct because they have different enzymes than we do. They can interfere with absorption of fat-soluble drugs and vitamins. Okay? Proteins are a different animal, right? Proteins are the workhorses of life, and proteins are made of building blocks called amino acids, and most of our protein comes from meat, okay? Some of it comes from vegetables and fruits, but not that much, okay? So I've said repeatedly that we are what we eat. A lot of us are basically, a lot of our body mass is basically meat. And so if we eat meat, we have all the components to make our human meat, right? Our human muscle and our other human proteins. So dietary sources are meat and dairy, okay? There are proteins in vegetables, but you have to be careful about the vegetables that you eat, right? You have to mix and match, for instance, grains and legumes in order to get what we call complete protein, which is all of the essential and non-essential amino acids. If you're missing an amino acid in your diet, then you don't have the ability to build all the proteins in your body, and you'll have a nutritional deficiency. Legumes, nuts, and cereals have incomplete protein. Legumes and cereal grains together have all the essential amino acids. So if you mix and match those, then you get everything you need. An example would be red beans and rice, right? You got complete protein in that one dish, okay? Now, the main use of protein is to carry out really any life process that you can name. Structural proteins include collagen and keratin and elastin, all right, and actin and myosin, the muscle proteins. They act as enzymes to catalyze chemical reactions. Some of them are going to be hormones, and sometimes the amino acids can be utilized for energy, but primarily the reason to eat protein is to make new protein. All amino acids are needed in order to build the proteins in our body. If they're not all present, then we can't build those proteins that promote life. Protein is used as fuel if we don't have enough carbs or fat. So if you eat a balanced diet, your protein component is going to be primarily to make new protein. It's not going to be to generate energy. Okay, it's basically building blocks. But if you eat nothing but protein, like let's say you were doing an Atkins where you were just you cut out the fat and you went all protein, then your body would shift in order to try and convert some of that protein into keto acids so that you could continue to make energy. Okay. Your nitrogen balance is just the balance between your nitrogen intake and your output. Normally, those are equal. Okay. The rate of protein synthesis usually is going to be equal to the rate of protein breakdown and loss. In a positive nitrogen balance, synthesis exceeds breakdown, and we see that in young children who are growing, okay, in pregnant women because they have the baby that's growing, and we see this in tissue repair, such as, let's say you have a burn or an abrasion, or you're recovering from trauma, okay. A negative nitrogen balance is where breakdown exceeds synthesis. Examples includes stress, infection, injury, poor dietary protein, or starvation scenario. Okay. One of the reasons, for instance, that you see the typical body shape of starving children in third world countries with those stretched out bellies is not because they're fat, but it's a condition called ascites, in which fluid collects in the abdomen because the liver can't make sufficient albumin in the blood for it to hold on to its fluid balance. And so the result is that you leak fluid into the abdomen because the peritoneum, which has a lot of blood vessels in it, loses that fluid to the surrounding tissues. Okay? And that would be an example of an individual with a negative nitrogen balance. In this case, it's caused from an imbalance in the diet. Okay? Hormonal controls, anabolic hormones such as growth hormone, testosterone, estrogen, accelerate protein synthesis and growth. Anabolism, remember, is to build large things from tiny things. Glucocorticoids promote protein breakdown and the conversion of amino acids into sugars, and that raises the carbohydrate level in the blood, the glucose level. You can see the total protein needs here in this pie chart were broken down into each of the individual amino acids. These are the essentials here, right? And you can see how when you combine legumes and grains, you get complete protein, right, for the essentials. The others, of course, you can build from alpha keto acids because of the action of the liver, okay? So those are our protein needs right there. Dietary requirements reflect our age, our metabolic rate, and our nitrogen balance. Rule of thumb is about a gram per kilogram of body weight. The American diet has way more than that, which is not a surprise. Vitamins are organic compounds that function primarily to help enzymes carry out their chemical reactions. Okay? Vitamin D is synthesized in the skin along with help from the liver and the kidneys. B and K are made by our intestinal bacteria in our colon. Beta carotene, which we get from our yellow and orange vegetables, is converted into retinol, which is used in the rods and cones in the retina of the eye in order to convert light into electrical information that we interpret as vision. Okay. The rest have to be ingested. Okay. So we have to eat them to get them, and no one food group is going to have every vitamin in it. Okay. Now, water soluble vitamins are the B complex and C vitamins, and they're used primarily as coenzymes. All right. B12 is a special case because its absorption across the lining of the stomach requires a protein called intrinsic factor. None of these are stored in the body because they're water soluble. If you take high doses, you're just going to end up putting them in the urine and you're going to pee them out. Okay, so mega dosing on the water solubles really doesn't do you any good. The fat soluble vitamins are a different case. Okay, A, D, E, and K are going to dissolve in our lipid stores and they're going to pass across the phospholipid bilayer with ease. Okay, they're stored in the body with our adipose tissue, with the exception of vitamin K, which again we get from our colon bacteria. But you can end up producing diseases if you have too much. Okay, um, any fat soluble compound has the ability to be almost stored in infinite amounts in the body, and that can produce danger, especially if you get to toxic levels. Some examples of this, back in the 1800s, the job of chimney sweep, where you had to clean the ash out of the chimney so that you didn't block the smoke exiting the house, um, had associated with it an occupational risk, which is that the burning coal produced a compound called diethylstilbestrone, which was a lipid soluble carcinogen. And so a lot of the chimney sweeps ended up dying of testicular cancer. And that's because the carcinogen was fat soluble and went easily through the skin and into the blood and then would affect various organs and tissues. Elemental mercury was once used to treat hat bands, and this was another occupation during the 1800s when hats were very popular, and the hatters that owned the stores that sold the hats would try the hats on, and the mercury from the hat bands would sink through their skin and into their brains and would cause problems with brain function, and that's the origin of the term "mad as a hatter." Okay, in the 60s, a fat-soluble drug, a hallucinogen called LSD, was utilized on sugar cubes and as a form called window pane, where you would put it on your eye, and it would go straight through the membranes and into the brain, and you would have hallucinatory experiences. And what a lot of people didn't realize about these drugs or these toxins is they collect in our adipose tissue, and they can recycle. Okay, even after you no longer using the drug, and can re-exert their effects. And that's why a lot of people that um, dropped a lot of acid in the 60s ended up having flashbacks later, is because those fat soluble compounds were actually still in their system. Okay, um, a caveat here, or a warning perhaps, to a lot of people listening. There are also fat soluble compounds in tattoo ink. Okay, and we don't yet know the effects of that. So just a cautionary tale. Fat soluble stuff is dangerous. Another example of a fat soluble compound is lead. Okay, vitamins have a variety of uses. Coenzymes is one of the major ones. They also can destroy free radicals that are produced during metabolism. Free radicals are basically electron poor molecules that steal electrons from healthy molecules and break them down and destroy them. Vitamin C, E, and A, and selenium are antioxidants that neutralize these free radicals. Where do we get them? Broccoli, cauliflower, Brussels sprouts for A and C. Okay. Minerals are required in moderate amounts, and examples include calcium, phosphorus, chlorine, magnesium. Others are required in smaller amounts, such as iron and manganese. But just because they're required in trace amounts doesn't mean they're useless, right? If I removed all the iron in your body, for instance, you would die because you wouldn't be able to make ATP and you wouldn't be able to carry oxygen in the blood because iron is at the core of both of those processes. So even though you don't have to eat tons of iron, you do need some in order to support normal function. The minerals work with nutrients in order to help the body function. A lot of them aid enzymes in performing their chemical reactions. Uptake and excretion are normally balanced to prevent toxic overload. Most minerals are water soluble. Okay, so if you take a large amount of those minerals in, generally, if you don't do it at an absurdly high rate, your body will eliminate those minerals before they build up to toxic levels. When you have a mineral imbalance, it's generally either because you've really OD'd on that mineral to an extreme extent, but primarily it's often due to a defect in hormone levels that control um, ions in our tissue fluid, okay? Or other disease states, things like trauma or certain infectious processes can do this as well. Examples: calcium, phosphorus, and magnesium salts harden bone. Iron is critical for carrying oxygen in the body as a component of hemoglobin. Iodine is critical for thyroid hormone synthesis. And sodium and chloride and potassium are important in establishing membrane potential in the body. Mineral-rich foods include vegetables, legumes, milk, and meat. Okay, metabolism we've actually touched on before. Um, Remember that this is the sum total of all the chemical reactions that support life, and we can break it into two broad categories. Anabolic reactions, where we take small molecules to make big ones, and this is a process that normally requires energy in order to proceed. So we would say chemically that these reactions are not spontaneous because the energy content of the products is higher than the energy content of the reactants. So you have to draw energy from somewhere else. And then catabolic reactions, where we take large molecules and bust them into tiny ones, and then take the energy that comes out in order to use that to support anabolic reactions, right? So the catabolic reactions make the anabolic reactions possible. Respiration is just the breakdown of the fuel that we take in, taking the energy from that fuel and using that to generate ATP. The net equation is glucose plus oxygen yields carbon dioxide and water. All right, that would be one glucose molecule plus 6 oxygen molecules gives us 6 CO2 molecules and 6 water molecules. That's the balanced equation. The other components there are that we produce about 38 ATP in that process, and we consume 38 ADP and phosphate, and we also produce a significant amount of heat in that process as well. Okay. The enzymes shift high-energy phosphate groups of ATP to other molecules in a process called phosphorylation, and these molecules are then activated and perform cellular functions. This is part of what's commonly called cell signaling, right? The phosphate groups are like turning on a light switch, and once we do that, the enzyme is ready to actually go out and do its job. Okay. In processing nutrients, we have digestion, which we talked about in the digestive chapter, absorption, which is the movement of those breakdown products into the bloodstream across the lining of the digestive tract. That would be for the amino acids and the simple sugars and the water-soluble vitamins, and it would be into the lymphatic system for the fats, okay, into the lacteals, as you remember, which are embedded in the villi of the small intestine. And then we route that stuff. We route the stuff in the blood to the liver first, okay, and then the liver stores it, and then when the tissues need it, the liver will free those things up and let the rest of the body access that material, okay. And in that form, it can move it across the membrane and use it to do work. Cellular processing of this takes place in the cytoplasm. This is where we build lipids, proteins, glycogen, and we break down glucose into pyruvate and convert that into acetyl-CoA as it passes into the mitochondria, and then it enters a process known as the Krebs cycle. And then the mitochondrial breakdown of the products of glycolysis to ultimately produce CO2, water, and ATP, right? And that's that net chemical reaction we just discussed. The goal is to move the energy to ATP so that the cell can use it to do work. It also generates heat and maintains body temperature. And we also store some of this energy, especially if it's excess energy, in glycogen, which is found in liver and skeletal muscle, and in fat, which is found throughout the body. We oxidize this food for fuel. Remember that oxidation is essentially the loss of carbon-hydrogen bonds from a fuel molecule, okay? And it's always going to be accompanied by something else being reduced, okay? And so it's a series of oxidation reduction steps that strip the energy out of the carbohydrate, and ultimately it gets passed to ATP, okay? So once the carbon components have been completely oxidized, the last step is the production of CO2, right? And there's no chemical potential energy in CO2 for living systems because there's no carbon-hydrogen bonds in CO2. The three legs of metabolism are glycolysis, the Krebs cycle, and oxidative phosphorylation, or sometimes we call that the electron transport chain, okay? So you can see the flow chart here, right? This is to give you an overview of what goes where. If we take a carbohydrate, right, we're going to turn that into simple sugars. We're going to send that into the blood. The blood's going to ultimately deliver it to the tissues. The glucose will be transported into the cells, and then enter glycolysis. Pyruvic acid will be converted into acetyl-CoA, which will enter the Krebs cycle, which will kick out CO2 and generate high-energy molecules like NADH and FADH2, which will then donate high-energy electrons to the electron transport chain. And then as those electrons move down the chain and ultimately get donated to oxygen to form water, we use the power that is stolen in the chain from those electrons to generate a proton gradient across the inner mitochondrial membrane that is ultimately used to synthesize most of the ATP in the breakdown process. Okay, glycerol can be converted into pyruvate. Fatty acids can be converted into acetyl-CoA. Right, amino acids can be converted into pyruvate or into acetyl-CoA, and they can also sometimes directly go into the Krebs cycle through a process called transamination or deamination. Okay, so those are some of the different ways that we can pass these various fuel molecules into the pathways that generate the energy. Now the anabolic pathways are here in purple, right? Glycerol and fatty acids can be combined to make triglycerides. Amino acids are used to make proteins, and glucose and other sugars are used to make glycogen, right? Which is the minor fuel storage molecule in the body. In an oxidation reduction reaction, what we have is the movement of electrons from the molecule being oxidized to the molecule that's being reduced. As a result, the molecule that's being oxidized ends up with less chemical potential energy than the molecule that's being reduced. Okay? And so oxidation is an energy stripping process, and reduction is an energy gaining process. Now, how do you tell if an organic molecule is oxidized or reduced? Okay? And the answer is very simple. You just count the number of carbon-hydrogen bonds. Okay? So for a a carbon-based molecule that has a certain number of carbons in it, like 3 carbons, okay? If you have, let's say, a total of um, 8 carbon-hydrogen bonds, all right, that's a highly reduced carbon molecule. That's actually a molecule that's called propane, okay? But if I remove some of those carbon-hydrogen bonds and form um, propanol, for instance, or propanone, which is a ketone, um, what happens is that that molecule has lost some chemical potential energy because it has fewer carbon hydrogen bonds, and that energy went somewhere. Okay, the ultimate oxidation of that molecule would be as 3 molecules of CO2. Okay, and then there'll be no chemical potential energy left in those 3 carbons that were once connected to each other. Okay. So that's an easy way to determine if an organic molecule is oxidized or reduced. Now, chemists would give you a different definition. A chemist would say that an oxidized molecule is one that has a positive change in its oxidation state, and a reduced molecule is one that has a negative change in its oxidation state. And there's a variety of rules that govern how you assign oxidation states. And that's beyond the scope of this course, but it is part of what we call general chemistry. Okay, so if you guys take Gen Chem, that's the definition that they'll use. Okay, a lot of these reactions are catalyzed by enzymes. Enzymes are generally named for the reactions that they catalyze. Examples include dehydrogenases, which remove hydrogen atoms from molecules and thus oxidize them. Oxidases, which move oxygen from one molecule to another. A lot of these require coenzymes to work, such as B vitamin derivatives. The coenzymes can act as hydrogen or electron acceptors. Examples include niacin derivatives, NAD, and flavin, which is B6, right? And that is FAD, okay? And both of those, when they're reduced, NAD becomes NADH and FAD becomes FADH2. And those are used to shuttle high energy electrons to the electron transport chain from the Krebs cycle, which happens in the mitochondrial matrix. There's 2 methods of ATP production in the 3 legs of metabolism. In substrate level phosphorylation, high energy phosphate bonds are transferred from one organic molecule to ATP, to make ATP from ADP. Okay? And that happens a few times in the 3 legs. It happens in the Krebs cycle and it happens in glycolysis. Okay? In oxidative phosphorylation, we generate a proton gradient in order to produce ATP by oxidizing NADH and FADH2. And then that proton gradient is relieved as those protons flow from the intermembrane space between the inner and outer mitochondrial membrane through ATP synthase in order to manufacture ATP. Okay? So high energy phosphate groups are directly transferred from phosphorylated substances to ADP, right? Happens twice in glycolysis and once in the Krebs cycle. Its necessary enzymes are in the cytosol for glycolysis and the mitochondria for the Krebs cycle. And it's kind of important, we should address essentially where these things are happening, right? Glycolysis is where? In the cytoplasm of the cell. Okay, all the enzymes for it are there. The electron transport chain happens where? In the inner mitochondrial membrane, okay, in the cristae, okay? And then the Krebs cycle happens where? In the mitochondrial matrix, okay? And the proton gradient produced by the electron transport chain happens between the inner and the outer mitochondrial membrane, okay? So that's where the protons get pumped into that inner membrane space. We've talked about enzymes before. Enzymes are specific for a particular substrate, and they catalyze a chemical reaction without being consumed, okay? You're looking here at substrate level phosphorylation, where we take a high energy phosphate bond from a substrate, we take ADP and we transfer directly and we generate an ATP as a result, and then we let go of that substrate that has now been reduced in its energy content. Okay. In oxidative phosphorylation, again, the process is as we've described, right? The energy from high energy electrons of NADH and FADH2 is stripped out as those electrons flow down the chain, and then pumps will move protons from the matrix into the inner membrane space, generating now a pH gradient across the membrane. Okay? And then what happens is that that pH gradient is relieved as it flows back through ATP synthase in order to make 32 of the 38 ATP that you can produce in running all three legs of metabolism. So you can see here, right, the pumping from matrix into intermembrane space, and then the electrons flow back through ATP synthase, and that is literally current, right? Charged particles moving in a direction down their electrochemical gradient, and then that drives the movement of this little rotor, which makes the ATP. That's the net equation I was talking about earlier, right? Glucose plus 6 oxygen for 32 ADP plus 32 phosphates give us 6 waters, 6 CO2s, and 32 ATP and a buttload of heat, all right? But that equation doesn't happen all at once, right? It happens in many, many steps. Otherwise, too much heat would be produced and you would cook the organism. And so that's the reason that we do it piece by piece by piece. So the three pathways are glycolysis, the Krebs cycle, and the electron transport chain. And we talked about where these things happen, right? So you can see where they're going on here, right? You've got the cytoplasm for glycolysis, right, where we take glucose and generate pyruvic acid and some ATP and some NADH, right? And then the pyruvate moves across the mitochondrial membrane, gets converted into acetyl-CoA in the process, and then from that we make some NADH and some ATP. And then in the Krebs cycle, we generate some ATP directly, but mostly we generate NADH and FADH2, and that ends up being oxidized by the electron transport chain in the inner mitochondrial membrane in order to make 32 of the 38 ATP that you get from completely oxidizing a molecule of glucose. An important point about all this, right, is that the electron transport chain will not run if there's no oxygen around, because the oxygen is what we call the terminal electron acceptor, and it's what picks up the low energy electrons and then leaves as water, okay, and that allows the chain to continue to flow. If there's no oxygen, or if there's something besides oxygen at the end of the chain, then the chain will get plugged up with electrons and won't work. And the result is we won't make any ATP. Okay? So when we start glycolysis, glucose comes into the cell through the sodium glucose synporter and is immediately converted into glucose 6-phosphate. And the reason for that is to trap the glucose inside the cell so it doesn't flow out the other direction. Okay? Most cells lack enzymes for stripping the glucose, the phosphate off of the glucose, and so the glucose is now locked into the pathway, right? In the intestine, the kidney, and the liver, the reverse reaction can occur, which means those tissues are capable of gluconeogenesis, all right, because they can go the other direction, all right? But that happens at different times and in different locations in those cells than in the locations that carry out glycolysis, okay? So those two processes are not happening in the same physical space, okay? They're separated from each other. So this keeps intracellular glucose concentration low and allows glucose to continue to flow through the synporter, right? Glycolysis is a 10-step pathway, and it is anaerobic, so it can take place regardless of whether oxygen is around. It's in the cytosol, and it ends up generating 2 pyruvic acid molecules. It has 3 major phases: activation of sugar, splitting of the sugar, and then oxidation of the sugar and the formation of ATP. Okay? And there's also some NADH formed in the process as well. In the sugar activation stage, Glucose is phosphorylated by 2 ATPs to make fructose 1,6-bisphosphate. This is an energy investment phase and provides activation energy for the reactions that come later. In sugar splitting phase, fructose 1,6-bisphosphate generates two 3-carbon fragments, DHAP and PGAL, okay, dihydroxyacetone phosphate and phosphoglyceraldehyde, okay. And then the following part is the oxidation of the sugar, okay. It has two major events. The 3-carbon fragments are oxidized and generate NADH. An inorganic phosphate group is attached to each fragment to make something called phosphoenol pyruvate, all right? And the phosphoenol pyruvate can undergo a substrate level phosphorylation of ADP to make ATP, okay? And so that is where one of the locations where it happens in glycolysis, okay? So you can see, again, the pathway here, right? So you've got the overall pathway up, all three pathways up top, and then we follow glycolysis down. This is all in the cytoplasm, right? We end up taking in the sugar, okay? The sugar gets split into two 3-carbon molecules. We end up generating two NADHs and 4 ATPs while investing 2 ATPs up top. So you have a net gain of 2 ATPs directly here. You got 2 NADHs that you're going to oxidize later, right? And then you're going to send the pyruvates into the Krebs cycle, okay? The final products are 2 pyruvates, 2 NADHs, a net gain of 2 ATP. In order for glycolysis to continue, you got to have NAD around in order to accept hydrogen atoms and continue the early phases of the pathway. The supply of NAD is a limiting reagent. NADH has to donate accepted hydrogen atoms to become NAD again. That happens at the electron transport chain, and that lets glycolysis continue. If oxygen is present, the chain can keep doing this over and over again, but if there's no oxygen around, then what will happen is it will go into something called fermentation, in which we will reduce pyruvate to lactate, regenerate NAD, and continue to run glycolysis because we really can't run the electron transport chain, okay? And that is basically a stopgap measure to generate energy in a pinch. Lactate will leave the cell and go to the liver and may end up being converted to glycogen, or may end up in glucose going into the blood, or may come back to tissues like muscle in the form of pyruvate and then have the rest of the energy stripped out of it as the muscle recovers from a phase of exertion. Okay? Prolonged anaerobic metabolism is going to generate an acid-base imbalance. The reason we go to glycolysis is because ATP production here is quicker than in aerobic respiration, even though the ATP yield is way less. So this isn't something that we can sustain for very long for two reasons. We're not getting enough ATP out of the process, but also we're creating chemical imbalances, all right? And this is generally why if we go into anaerobic respiration, after a short period of time, activity will stop in that cell or tissue, okay? So let's take a look at an overview of glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3 carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, the next phase in the 3 legs of metabolism is the Krebs cycle, and this happens in the mitochondrial matrix and begins with the conversion of pyruvic acid to acetyl-CoA and fatty acids in a stepwise fashion into acetyl-CoA. Okay? And it's in this form that they condense with a molecule called oxaloacetate and form the first molecule in the cycle, which is called citric acid, which is why this pathway sometimes bears the name of citric acid or TCA cycle. Okay? The transitional phase converts pyruvate into acetyl-CoA. There's decarboxylation, which is a highly energy releasing reaction. Okay, so it's an exergonic reaction. We remove a carbon and produce acetic acid and CO2. Hydrogen atoms are stripped from the acetic acid and make NADH. Okay, and we form acetyl coenzyme A as a result. Coenzyme A takes acetic acid into the Krebs cycle, where it combines with oxaloacetic acid to make citric acid. During 8 steps, the citric acid is decarboxylated, so meaning we strip out CO2s, and oxidized and generates keto acid intermediates and NADH, some protons, and FADH2. And in the final step, we regenerate oxaloacetic acid, and the cycle starts all over again. Okay? With each turn of the Krebs cycle, we produce 3 NADH, a proton, 1 FADH2, 2 CO2s, and 1 ATP. Okay? Now, to do the math on this, all right, each NADH equals about 3 ATP in the electron transport chain, and each FADH2 equals 2 ATP on average. Okay? Those aren't exact numbers, but on average. So 3 times 3 gives us 9, right? 1 times 2 gives us 2, so 9, 10, 11, right? And then 1 ATP gives us 12, right? And then we turn the cycle twice because we're getting 2 pyruvates to go in, and that gives us a total of 24, okay? And then you have to remember that we generated 2 NADHs in glycolysis, okay? So that's another 6, so that's 30, right? And then we generated 2 ATP directly in glycolysis, so that's 32, okay? And then we had 2 turns of the Krebs cycle, and we generate ATP directly from those, and that ends up giving us a total of 36 to 38, okay? And the variation is due to the efficiency of the electron transport chain, among other things, all right? So, there it is, right? That's the net equation. And there we go. We end up adding all those together, right? So 8 times 3 is going to give us 24, right? And then 2 times 2 is 4, that's 28. And then the 2 ATP for 30, okay? And there's another 6 in there as well, okay? So the Krebs cycle doesn't directly use oxygen. The NADH has to be oxidized by the electron transport chain. If not, then again, there's going to be a limiting reagent problem. So eventually, the Krebs cycle would shut down if there was limited oxygen available for an extended period, right? Breakdown products of fats and proteins can enter the cycle, and the intermediates can be used for other reactions, like to make amino acids to build proteins, as one example. The whole cycle is here, all right? But we'll look in detail at each step in the following video, okay? So let's consider that now.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, now with the electron transport chain, what we're doing is actually generating the bulk of the energy that is going to be produced when you add up all 3 legs of metabolism. It generates 32 of the 38 ATP that you get from completely oxidizing a glucose molecule. And in this process, NADH and FADH2 produced from the Krebs cycle donate high-energy electrons to the chain at different locations. NADH donates at the very beginning of the chain and FADH2 a little bit further down. And then as the electrons flow down this chain, which is embedded in the inner mitochondrial membrane, they lose potential energy, and that energy is used to pump protons from the matrix into the inner membrane space. And then the production of ATP is ultimately the result of the proton gradient diffusing through a membrane protein in the inner mitochondrial membrane called ATP synthase. And so as that current flows, as those protons go through the core of that membrane protein, it actually turns a rotor, and that kinetic energy is used to drive the synthesis of ATP from ADP and phosphate. In the chain, we have flavins that come from riboflavin, which is one of the B complex vitamins. We have cytochromes, which are iron-containing pigments. And we have respiratory enzyme complexes that pass the electrons from high to low energy states. Okay? And all of this happens with oxygen at the end of the chain to accept the low energy electrons, bind a couple of protons, and leave as what we call metabolic water. Okay, so as long as there's oxygen around, the chain will run. Okay, so these high energy molecules, NADH and FADH2, once oxidized, are going to return to the Krebs cycle and allow the cycle to continue to run. The protons initially form in the matrix and are pumped into the inner membrane space, while the electrons go directly into the chain and power the proton pumping capability of the chain until they get all the way to oxygen. There's 3 enzyme complexes, 1, 3, and 4. Okay, the protons create an electrochemical gradient that diffuses through ATP synthase, and that's what generates the bulk of the ATP produced, 32 of the 38. Okay? The oxidation of NADH and FADH2 generates sufficient energy to drive the production of all of this ATP. The electron pairs become water once they combine with the oxygen that's bound in the last complex. So you can see here, NADH donates at the top of the chain, flows from complex 1 to Q to 3 to cytochrome C to 4, and then we pump the equivalent of 3 protons, which is sufficient to power the production of 3 ATP as a result of oxidizing NADH. FADH2 donates further down the chain and gets the equivalent proton pumping power to equal 2 ATP at ATP synthase. Okay? The gradient accumulates in the intermembrane space. The only place it can be relieved under normal circumstances is through this hole. And so that's why oxidative phosphorylation works, because it's the current flowing through this little rotor that drives that kinetic energy to produce the ATP. So this is another energy transformation process that also produces a significant amount of heat. You can see the drop in in potential energy as the electrons flow down the chain to oxygen. Okay? And this is just a result of a shift in the position of the electrons. They're changing their location, so they're changing their potential energy, so they're changing their energy content. It's a lot like potential energy in the world you live in, right? If you take the elevator to the top floor of a building, you have more potential energy on that floor than you did on the bottom floor. And you could turn that potential energy into kinetic energy by jumping out the window. If you jumped out the window on the first floor, there would be less kinetic energy produced. Okay? So it's the same thing here. The electrons change their location. As they change their location, they change their energy content. The energy lost is used to produce that proton gradient across the intermitochondrial membrane. Okay? ATP synthase has 2 parts, the rotor in the intermitochondrial membrane and the knob which is in the matrix. And what happens as current flows through it is that we turn the rotor. The knob is where the ATP synthesis is located. This also allows ADP, pyruvate, and inorganic phosphate to cross the intermitochondrial membrane and make it into the matrix. And this is what it looks like in an electron micrograph. So you're looking at the matrix side of ATP synthase here. Okay, kind of cool. So you can see the mechanics, right? As the protons flow down their gradient, we turn the rotor. The knob is going to utilize that current to generate ATP from ADP and phosphate, and there's going to be heat given off, and that generates 32 of the 38 ATP that's produced by all 3 legs of metabolism. So let's take a closer look at the electron transport chain.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD plus and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, so if we go through ATP production, the energy use is about 100 kcals per hour, which is a big demand on the mitochondria. A mole of glucose, thus, is going to produce 686 kcals, trapping about a third of that as ATP, with the rest lost as heat, which is 40% efficient, which is pretty good because our best machines are 30% efficient. Okay. Complete oxidation of one glucose molecule from glycolysis to Krebs cycle to electron transport chain is going to give us CO2 and water and 38 ATP. I know it says 32 there, by both substrate level and oxidative phosphorylation. The energy required to move NADH and protons generated in glycolysis into the mitochondria brings the total down to around 30, but the total is in debate. Okay, and everybody's total is going to be a little bit different, and you might wonder, well, why is that? Well, every enzyme in this chain is coded by genes, and there are variations in those genes that produce enzymes that have different efficiencies. The more efficient the enzymes of the three metabolic pathways are at doing their job, the more ATP you're going to produce. The result is you're going to tend to store more energy. Okay? The less efficient these enzymes are, the less efficient your ATP production will be, more of your energy will be lost as heat. You'll store less energy as fat. And so that's why some people are genetically thinner than others. Even if they eat the same amount of calories, they can lose more of it as heat as a result of the efficiency of the components in the chain. Now, the other variable, of course, is your exercise level, right? The more energy you burn by doing exercise, then the less energy you're going to store, okay? And that's the environmental component that dictates your weight, okay? So there's a genetic component, there's an environmental component. Environmental component is how much you eat and how much you exercise, and the genetic component are the genes and the DNA that you inherited that code for the enzymes in this pathway, okay? And so there's different flavors for everybody. So you can see here all of the production of ATP. Now, you've got to remember here, net gain of 2 ATP here, but remember that when we move acetyl-CoA into the Krebs cycle, we're getting a couple of NADHs here in addition to a couple out here, all right? So that's another 6 right there on the movement from acetyl-CoA into Krebs, right? So we can tack 6 onto their total, right? They're getting 2 here, they're getting 2 here directly, okay? And then they're getting around 30, all right? So they're getting 34, okay? And then 2 and 2 and 30, 34, and then we're saying another 6. So let's call it 38 on average. And then we burn some doing this work, right? And that brings the total back down. So the debate on the total is an ongoing thing, okay? Glycogenesis is the reverse process where we synthesize glycogen from glucose monomers, and this consumes ATP and happens only in liver and skeletal muscle and serves as a form of quick energy when the body needs, or when muscle needs some glucose real quick because the blood can't supply it fast enough. It can break down its glycogen. The same is true when blood sugar levels fall for a brief period. We can generate glucose and send that into the blood from the liver's glycogen stores. Glycogenolysis is the process of breaking down that glycogen, right? And so that's when we want to utilize that stored energy and put it into the blood. We take the glycogen, we split it into glucose, we let it go into the blood, and this happens only in liver cells, okay? And as a response to low blood glucose levels. So you can see that the pathways have similar intermediates, okay? But they happen in different cells and in different locations within those cells. And as a result, they don't interfere with each other. And that makes us more efficient at doing both tasks, right? And they also happen at different times, okay? So the timing is different, the locations are different, and frequently the tissues are different. And this way you don't end up with processes that do the opposite happening at the same time in the same place, right? And they would interfere with each other. Complex carbohydrates allow us to store more glycogen, and they're more effective than high protein meals for intense muscle activity. Carb loading is a carb-rich diet for about 4 days. Decreased activity allows the muscles to store more glycogen, and that increases the performance and endurance in those muscle types. Okay. Glucose formed in the liver from glycerol and amino acids when blood glucose levels drop is a process known as gluconeogenesis, and it protects against hypoglycemia, and that's critical for nervous system function, right? So in a starvation scenario, your body is going to start chewing up your protein and giving it to your central and peripheral nervous system so that you can continue to live, right? And so what you'll see there is muscle wasting. Okay? Lipids are the calorie champ, right? They have more calories per gram than any other nutrient. Fat breakdown is going to give you 9 kilocalories per gram versus 4 for carbs or protein. That's why if you eat a lot of fat, you don't do a lot of exercise, you're going to gain weight. Most products of fat breakdown end up in the lymphatic system in the form of little bubbles of fat called chylomicrons that have polar components projecting outward and the nonpolar fatty acid tails projecting inward. Okay? They're hydrolyzed by endothelial enzymes into fatty acids and glycerol, and then they can be used by the cells and tissues in the body. The triglycerides are oxidized for energy, okay? And then the building blocks are oxidized in different pathways. There's a glycerol pathway which goes directly into the Krebs cycle after passing through a few steps of glycolysis. And then the fatty acid pathway is going to directly generate acetyl-CoA units by stripping 2 carbon blocks off of the end of the fatty acid chain and donating them to oxaloacetate as the first step in the Krebs cycle. Okay, so glycerol gets converted to PGAL, phosphoglyceraldehyde, goes into the Krebs cycle after becoming pyruvate, right? And the ATP yield is about half of what you get from glucose because it's just 1 3-carbon molecule, right? So you get 15 ATP per glycerol. The fatty acids are different. The fatty acids are broken down in 2-carbon units. And what happens basically is you work from the acid end of the fatty acid back towards the end of the tail, all right? And you produce something called acetyl-CoA, okay? And you utilize sulfhydryl groups in this process, primarily a coenzyme called biotin, all right? The coenzyme, once it's reduced, is going to donate to the electron transport chain, all right? So you can see here how the pathways differ, all right? So the fatty acids are broken down in 2-carbon groups. We generate coenzyme A. The process is called beta oxidation, right? Because we start at the acid end of the chain, and then we oxidize a carbon that is 2 carbons away from that acid group. And then we strip that CoA group off of the fatty acid, and then that carbon that became oxidized, which has a keto group on it now, becomes the next acid group to become a coenzyme A, an acetyl coenzyme A. All right. And so what happens here is that we produce an NADH and we produce an FADH2 for every step. Okay. We end up feeding into the Krebs cycle as acetyl-CoA, and we get a single turn of the Krebs cycle out. So you can see that since a typical fatty acid is about 20 carbons long, you're going to get about 10 of these turns of the Krebs cycle, And then you multiply that by 10 of these beta oxidation steps. Okay? So you get tons of energy out of burning just one typical fatty acid tail. Now, a saturated fatty acid is going to give you more ATP than an unsaturated one because it has more carbon-hydrogen bonds, right? Um, and a polyunsaturated will give you less than a monounsaturated, and a saturated will give you more than either a polyunsaturated or a monounsaturated, right? Unsaturated fatty acids, you can tell because they have double bonds in them, all right? Lipogenesis is just the production of fat. Dietary glycerol and fatty acids aren't needed for energy, get stored as fat in the body, adipose tissue, triglycerides. And this happens when ATP levels are high. The glucose is converted into fat because acetyl-CoA is an intermediate in glucose breakdown and a starting point for fatty acid synthesis. Lipolysis is the reverse of lipogenesis. The stored fat is broken down into glycerol and fatty acids. The glycerol enters the back end of glycolysis and goes then from there to the Krebs cycle. The fatty acids get broken down by beta oxidation and become acetyl-CoA and enter the Krebs cycle that way. Oxaloacetic acid is important for complete oxidation of fat. It'll get converted to glucose if the carbohydrates are low in your diet. Okay, so this is how the Atkins diet works, right? The Atkins diet, by cutting your carb intake, cuts down on oxaloacetate levels, which means you can't run the Krebs cycle real well, and that's why that extra acetyl-CoA ends up becoming ketone bodies instead of entering the Krebs cycle. And then those ketone bodies are generally not metabolically useful. They're like metabolic dead ends, and they end up in your pee and your sweat and your breath. And that's how you basically vaporize your body fat on Atkins. The problem, of course, with that is that it sends you into ketoacidosis. That screws up your body pH, and that can end up being lethal, okay? Which is why you don't do it for long periods of time, okay? And so that's the ketogenesis and the ketone bodies that we were talking about. The ketones are acidic. They send you into metabolic acidosis. You get a fruity smell to your sweat and your breath and your pee. You breathe rapidly to get rid of CO2 in an attempt to try to raise your body pH. We see it in starvation, in people that misuse the principles of the Atkins diet, as an example. We also see it in diabetes, and the reason for that is in diabetes, you can't use the sugar in your blood for energy because you've become insensitive to your own insulin, or you don't make insulin. And as a result, your sugar levels in your blood skyrocket, and your body has to go to your fat primarily for fuel. And when that happens, you lose carbohydrate intermediates like those in the Krebs cycle, and you go into ketoacidosis. Okay, so you see this in the urine. Ketone bodies in the urine is called ketonuria. Okay, and you also get low pH in your urine as a result. And so you can see there the combination of all the different pathways that we can go in and out of when we're talking about fat, and whether we want to synthesize it or break it down, right? And keep in mind, again, that if we're building fat, we're using similar intermediates, but we're using different enzymes that work in different locations than those that are for the breakdown of fat. And that's why the two processes don't interfere with each other, okay? And they also tend to happen in different locations in the cell, okay? Now, lipids are used for phospholipid production, which is the major component of cell membranes. Cholesterol is for the control of membrane fluidity in the liver. Synthesis of transport lipoproteins for cholesterol and fat take place, okay? And synthesis of cholesterol from acetyl-CoA takes place. And this mostly happens in the smooth endoplasmic reticulum, which you may remember from A&P2. We also use cholesterol as part of bile salts, and the reason for that is to serve as a component of the biosalt that's a detergent, right? The biosalts have a nonpolar and a polar part to them. They are stored in the gallbladder where they become concentrated, but they're manufactured in the liver, and they serve to emulsify the dietary fat that drops from the stomach into the duodenum of the small intestine. Okay, and by that detergent action, what happens is that the large fat droplets get smaller, right? They get mechanically broken down in a process called emulsification, and that provides more surface area for the pancreatic lipase to chemically break down the fat so that it can eventually be absorbed into the lymphatic system as chylomicrons. Okay? So that brings this section of chapter 24 to an end. Make sure and pick up the back end in the next podcast, and I will see everybody in the next chapter. I thank you for listening.